In part 2 of this unit, we'll talk about proteins. So the last two biological macromolecules are going to deserve a part of the lecture in themselves. So we'll talk about proteins first. Proteins are super important biological macromolecules. They are essential in so many different functions in the cell, including structures. They form enzymes that we talked about. They are important for speeding chemical reactions in the cell. They form transport proteins that help move chemicals across membranes. They form structures like flagella that aid in the movement of bacteria. They form toxins for bacteria and other important cell structures. They do everything. Proteins are polymers that are made of monomers called amino acids. Right? So amino acids have this regular chemical structure that you'll see repeated in the protein polymer, where there is a carboxyl group, a carboxylic acid end. There is an amino group. Remember, amine is a keyword for nitrogen. And then there is a side group. So that chemical structure of amino acids looks like this. Right? So we have the amino group. That's the nitrogen-containing end. We have the carboxylic acid, or the carboxyl group end. Right? Those are joined together by a single central carbon that we call the alpha carbon. And now that alpha carbon, it's bonded on to the amine on one end and the carboxyl on the other end. Those are the two common functional groups. It's bonded to a hydrogen. And then it's got a single bond to a side group. So this R is an abbreviation we use in drawing chemical structures. That just means the rest of the atom, or the rest of the molecule, excuse me. So for amino acids, in each of the different amino acids that make up proteins in biological systems, that R group, or that side group, is different. For example, this is the chemical group that makes up the amino acid tyrosine. Now that R group, or that side chain, gives all of the interesting features to each one of the amino acids. There are 20 different amino acids that biological organisms use to make proteins. Right? The side chains of each one of those is different, and it determines the properties and the function of the protein. Now, each one of those 20 side chains is unique, but they fall into 4 general categories as to the general chemical properties of those side chains. There are non-polar amino acids. There are polar amino acids. Then there are charged amino acids, including acidic or negatively charged amino acids, or basic or positively charged amino acids. So these are the chemical structures of the 20 amino acids that make up proteins in all life on Earth. Again, these are grouped into these 4 distinct types of amino acid side chains. We have the non-polar side chains, and one thing you should definitely do after Unit 3, you should go through and recognize that these functional groups on these side chains, we have carbon and hydrogen, carbon and hydrogen, carbon and hydrogen, rings of carbon and hydrogen, rings of carbon and hydrogen with a single amine, and more rings of carbon and hydrogen. These are all non-polar and hydrophobic. Then we have these polar side chains. Again, look at those functional groups and their chemical properties, right? So here we have hydroxyls. Those are the polar functional groups. More hydroxyls. Here we have a sulfur, another electronegative atom. Here we have a ring of carbon and hydrogen, but also a big old hydroxyl on the end of it. And then we have some carbonyl and amine ends on asparagine and glutamine. Right? So those are all polar side groups. Then we have the charge. We have acidic side groups. Those are negatively charged because they are able to donate a hydrogen into the solution. Right? And then we have the basic side groups because they pick up a hydrogen, a proton from solution, in order to make them positively charged. So you have lysine, arginine, histidine, and then the acidic ones, aspartate and glutamate. Right? in the PDB video that you should definitely watch, it tells you a lot of important information about proteins and amino acids. They do mention that there are 21 amino acids. In fact, there are actually 22 that we know about. And so the 21st amino acid, selenocysteine, it is an amino acid. It makes up proteins in many organisms on the planet. We call this the 21st amino acid. It's weird and interesting. It includes one of these atoms that's not on the 6 that we actually need to think about. It's also put into proteins in a different way than these other 20 amino acids. So we're, for this class, mostly going to ignore it. If you want to know more about selenocysteine, you can ask me in class. So peptide bonds are the chemical bonds between amino acids that make up the polymer. Again, these peptide bonds, just like all the covalent bonds that make up polymers in these 4 macromolecules, they're formed by a condensation reaction where we have water removed when a covalent bond is formed between the carboxylic acid end, the carboxyl end, and the amine end. So that creates this carbonyl and amine in the middle of these 2 joined amino acids. So the peptide bond between amino acids forms these long chains of amino acids. So proteins are interesting not just because they have these long chain polymers of different amino acids that are joined together, but that those individual amino acids allow proteins to fold into interesting and complex chemical shapes. Right? So these 3-dimensional shapes give the proteins a lot of their important function. So we think about the 3-dimensional structure, the protein structure, in several different levels. We have the primary structure of proteins, and that is the sequence of amino acids that are joined together by peptide bonds. Right? Then those amino acids that are in that long chain, they can fold into 3-dimensional structures. Amino acids that are close together on the chain, they form hydrogen bonding interactions between those amine and carbonyl side groups. Right? And that allows them to form into 2 regular structures that you'll see in almost all proteins, either alpha helices, where we have a twist of a chain of amino acids, or beta pleated sheets, where we have sheets of chains of amino acids that are folded back onto each other. Those local structure, those secondary structure, then fold into a more complex 3-dimensional structure called tertiary structure. So the tertiary structure is the overall 3-dimensional structure of the molecule. And those tertiary structure interactions are primarily driven by the chemistry of those amino acid side groups. Then those proteins, once they're folded into 3-dimensional shapes, they can actually form interactions with other proteins to form what we call the quaternary structure. So that's when proteins stick together to form even more complex and interesting structures. So, primary structure of the protein, that is the chain of amino acids. Right? So those bonds between the amino acids on the chains, we call those peptide bonds. So the peptide bonds form the primary structure of the amino acid. Right? On one end of that amino acid, remember there's an amine and a carboxylic acid that participate in the peptide bond. So on one end of the protein chain, there's an amine that's not bonded to anything. And then on the other end of the carboxylic acid, there's or on the other end of the amino acid chain, there's a carboxylic acid that's not bonded to anything. So we have the N end, or the N-terminal end, and the C end, or the C-terminal end. Then each one of the amino acids in that chain, they consist of these 20 different amino acids, so they can be very different from each other. And the order of those amino acids, for example, phenylalanine, leucine, serine, and cysteine, the order is the basis of the additional chemical properties that we'll talk about in just a minute. So that's the primary structure of the protein. The secondary structure of the protein again consists of these 2 regular structures, either alpha helices or beta sheets. So the alpha helices are these twists of amino acids, and the beta sheets are the pleated sheets of amino acids. So alpha helices and beta sheets are made by interactions between amino acids that are close together on the overall primary structure of the protein. Right? Both of these are created by hydrogen bonding between the amine of an individual monomer on the chain and a carbonyl of an individual monomer on the chain. So those are those electronegative atoms that can form these hydrogen bonds that we'll talk about, especially when we talk about nucleic acids. All right, the hydrogen bonding between these nearby amino acids can form these regular structures where we form these beta sheets where those hydrogen bonds line up just so to form this sheeted structure. Or if those amino acids twist around each other, they also line up just right. So we have hydrogen bonds at each regular point on the twist. And that forms the alpha helices. So the tertiary structure occurs when the helixes and sheets fold into three-dimensional complex structures that are driven by lots of different chemical interactions between side groups and other parts of the protein. So we represent the structure of proteins using a lot of different diagrams. So here we have the three-dimensional structure of a protein represented by a diagram that shows these alpha helices and pleated beta sheets that are folded into this three-dimensional shape. Now, why do proteins fold into these regular three-dimensional shapes? It's because they have all these opportunities for chemical interactions between different parts of the protein. We can have ionic bonds formed between those acidic and basic groups, those acidic and basic side chains of amino acids, where two oppositely charged amino acids are able to form a strong ionic bond between them. We can have hydrogen bonding between two polar amino acid side chains. Right? We can have hydrophobic interactions where those hydrophobic amino acids, right, they don't like to interact with water, which makes up most of the biological environment. And so instead they stick to each other on the inside of the three-dimensional structure of the protein. We can also have structure in the protein made by covalent linkages between side chains. So the most important role for sulfur, that was that sixth atom that we told you to recognize, sulfur forms what we call disulfide linkages or disulfide bridges, where two sulfur-containing amino acids are able to form a covalent bond between those sulfurs on the side chain. And those create very strong, stable three-dimensional structures in the proteins. So, summary of the protein structure. We have the primary structure of the protein. That's the sequence of amino acids joined together by peptide bonds. We have the N-terminal end and the C-terminal end, and then the individual amino acids on the chain. We have alpha helices and beta sheets. Those are the short-range interactions between amino acids. And then we have tertiary structure, that is the complex three-dimensional folding of the protein overall. Then finally, we have proteins that interact with other proteins to form super complex structures, help proteins do all the different things they do. And that's the quaternary structure. So the structure of proteins is determined by the sequence and chemical properties of amino acids that make them up. And then that structure determines what the function of the protein is in the cell. A good example of that is hemoglobin, which is a protein found in your red blood cells that's responsible for carrying oxygen from your oxygenated lungs to your peripheral tissue where there isn't so much oxygen. Right? So what this oxygen carrier needs to do is that when it's in the lungs, when there's a high amount or high concentration of oxygen in those alveoli, right, oxygen needs to bind into, stick to this protein so that it can be carried to the peripheral tissue. Then when hemoglobin gets to the peripheral tissue, it has to somehow know to release the oxygen into that oxygen-depleted environment. Right? Hemoglobin does this by taking advantage of this three-dimensional structure of the protein. And so hemoglobin is actually made of several different proteins that come together to form that quaternary structure. So those chains of hemoglobin molecules, right, they are able to bind on to another molecule called heme, which is an iron-containing molecule that is really good at sticking to oxygen. Now the shape of the hemoglobin protein changes a little bit when it's in the highly oxygenated environment in the lungs, and when it's in the low oxygen environment in the peripheral tissue. That change in the shape of the protein in response to the chemical environment it's in allows oxygen to stick extremely hard to the heme in the oxygenated lungs, and then be released from the heme when it's in the unoxygenated peripheral tissue. That lets hemoglobin be a very good carrier for oxygen and deposit that oxygen around the body in the places that most need it.